It's a knife. No. Okay, hello. This is so unprofessional. But hey, it's for an assignment, so hi, I'm Maya, and today I will be doing an in-house experiment involving molecular biology. And this is it's so this experiment that I'm going to be doing is for the molecular biology lab assignment. That's what this is going to be for. So before we get into the experiment and what I'm going to actually do, I'm just going to be explaining that all of us, we all have DNA, okay? Our DNA is different from each other. Even with twins, although they look similar, some of their DNA is not the same. Some of them are because, they look identical. Why am I talking about this? Because by we, I mean me, gonna extract DNA, especially from bananas. Banana. Forget the half eaten. What are you talking about? This is a full banana stump, okay? No one ate it before, okay? This is a full banana stump. I'm going to be extracting DNA from it because guess what, girl? Not only is it easy to mush, the reason why I'm using banana is because it's easy to mush, it's easy to destroy. Whoa. It's easy to, destroy. The cells are easy to destroy. And also, fun fact, if y'all don't know, bananas apparently are we have kind of similar DNA. Like, there's a 90% chance, like something like that. I'll pop up on the screen. Like, we have a high percentage similarity of DNA with us Homo sapiens and bananas. So I guess we're cousins. Is it considered cannibalism if I eat it? I don't know, but well, let's get into it. I'm gonna kill my cousin today. For this DNA extraction process that you can do at home, by the way, yeah, you can do it at home. Um, I have pre-chilled the alcohol that I'm going to be using, but in this case, I'm going to be using hand sanitizer because the only alcohol that is in this household is for drinking, and I don't want to waste that. I don't want my dad to whoop my ass. So the alcohol that I'll be using is the hand sanitizer that all of us started, I don't know, bulk buying during COVID. Do we remember that? Do we remember that? I hope we do because that's insanity. But the thing is, it's been, I don't know, girl, 7 years? Oh my god. I don't know if this hand sanitizer is going to be working well, but in theory, it should because I helped my junior with the same experiment. I'm not copying her. Maybe I am. This is the only experiment that came to my mind when I saw the assignment, okay? Do not judge me, please, please. Hello, Professor. I know you're seeing this. Love you. Please do not put this for everyone in our class to see because I'll be so embarrassed. I pre-chilled the hand sanitizer, and I will get to you later on why we need to do that. The colder it is, the better because our DNA is very fragile. Okay, why am I doing this experiment? My objective is to isolate and I'm gonna visualize as in like try to extract the DNA. Like it's gonna be in clumps. It's gonna be in clumps. Okay, if my hand sanitizer is well, it's gonna be in clumps, and it'll be easier to pick using a toothpick. This will obviously demonstrate key molecular biology processes such as like cell lysis, cell breaking, and also DNA precipitation. The materials that we need are banana, our cousin, our ancestors over here. We also need soap, dishwashing liquid, but I'm going to just use a hand soap. We need salt and then water, and then we need the hand sanitizer. If you have alcohol like ethanol, um, but other like just like coal alcohol, like I don't know, girl, only ethanol comes to my mind. I'm gonna be needing bowl, spoon, and I'm gonna filter. So I need cloth or tissue, but I think I'm gonna use tissue. And then I need a cups, plenty, and also toothpicks. So first step that I'm gonna be doing is that I'm gonna be mashing my ancestors. I'm gonna break them apart. Damn. The amount of DNA depends on how much bananas you're taking and the size of the banana, right? This is quite small. It's gonna be smushing the banana. Okay, see how mushy that got? Yep. Yeah, you need it as mushy as you can. Trying to make it as mushy as possible. Now that I have mixed enough of the banana that is like mushy, yeah, that's the consistency that we want. So what I did was breaking open the cells, right? So instead of like being a banana shaped, it's now in this like liquidy slash clumps form. So now what I'm gonna do is that I'm gonna be preparing an extraction solution. So what is this? I'm going to be mixing water, salt, and the liquid, the detergent, which I'm gonna be using hand soap, and this is gonna be the magic solution where the detergent will actually help dissolve the membrane of the cells and the nucleus. And the salt that we added is going to neutralize the phosphate backbone, which is negative charge, um, phosphate backbone of the DNA that is negatively charged, and this is going to allow the DNA to clump cups to prepare the solution. What I'm going to do is that I'm going to be taking a little bit of salt, maybe like a spoonful, so like maybe like this much salt, just like barely covering the surface of this plastic cup. I got my solution here, but I'm gonna mix it. It's gonna be, make sure to have as little bubbles as possible. So this solution will allow um, the extraction of the DNA much easier. Like I said before, the salt allows the clumping to occur. The hand wash is going to help dissolve the membrane of the cell and also the nucleus. I don't know if you can even hear me from how loud this is. ASMR. I'm going to be adding this salt water, this extraction liquid that I've made using water, tap water, salt, and also hand wash. And I'm going to be slowly adding to this DNA that I've mushed. DNA, my ancestors that I've mushed. And I'm going to slowly stir it. I'm going to be mixing it. What you gotta do is that you gotta slowly mix it, gently mix it for 2 to 3 minutes. Yeah. So we are going to be leaving any like huge chunks that we have here. You only need the liquid part, right? So what I'm gonna do is that I have another glass here. So what I'm gonna do is I'm gonna take my tissue. I'm gonna take my tissue and I'm gonna put it over on top of this glass so it can act like a filter, and all the chunks will be filtered out. See, that's the clear liquid that we want. This one, however, you can throw it away. We just gotta put alcohol because that's the thing that's gonna make it all the difference. All right. So I have the alcohol. It's in the freezer. It's in the fridge. So I'm gonna take it out and I'm gonna slowly add the alcohol to the sides and not in the center or just straight up pumping it, because the alcohol is gonna be what makes the clumping effect. So there's a layer between there. Oh, there it is. It's really hard to see here, but I'm just gonna let this sit for 3 minutes to 10 minutes. While we are waiting for this, I'm gonna be explaining. To summarize it, we mash the banana, we break open the cells. The cell experience cell lysis, and all of its components is coming out. Okay, and then we added the detergent, soap. This will dissolve the cell and also the nuclear membrane. And then we add the salt. The salt is going to help the DNA clump together and neutralize the phosphate backbone, the negative charged phosphate backbone. And then finally we added the coal ethanol, but in this case I used hand sanitizer. Why? Because it's the only thing that I have in my house and I don't want to buy alcohol separately because I'm not going to use it for anything else, nor are my parents going to be. So this hand sanitizer, you can also use ethanol or isopropanol, but I used cold hand sanitizer, and this is going to precipitate in DNA, and there's going to be a visible white um white strands that look like saliva, but it's like white strands. Here's the part, the interesting part. Why do we need to make the alcohol cold, in this case, like the hand sanitizer cold? Well, thank you for asking. That's a good question, actually. DNA is very fragile, very, very fragile, girl. Especially when you extract it, it's very fragile. Even in labs, we have to use it. We have to store the DNAs like negative temperatures. We need to use like a cold storage, okay? Cold storage. The coldness of this hand sanitizer is going to help DNA be less soluble. So it's not going to be mixing it easily and it'll be able to extract it easily. And then it'll also precipitate easy. Coldness allows it to precipitate easy, and also it will protect from degradation. Yes, degradation, we do not want that to happen. If we are trying to extract DNA, we just want to see, we don't want it to degrade, because if you pull it out, it's gonna be no strands. Imagine I get no strands later, even though I still use gold. Okay, that's the common principles on why we use certain things. This is the limitation. Like I previously mentioned, I did not use ethanol or any alcohol. Ethanol, yeah, or any alcohol. I used hand sanitizer. So in this case, in this experiment, when I used hand sanitizer instead of alcohol, the additives that they added into the hand sanitizer, yes, I know there's more in hand sanitizer. It's shocking, right? But things like glycerin and like gel that are added to make this product even more stable is going to reduce the efficiency and also the amount of DNA that we are going to be precipitating. So to better get better results, to get better results, to get more DNA, it's bestly advised to use alcohol if you have alcohol like ethanol or isopropanol. But if you do not, you can just use hand sanitizer, because after all, this is an in-house experiment. You're trying to use in-house things that can allows you to make you feel like you're a scientist. But in this case, I'm actually a scientist, and I have to prove that this actually works. So, what DNA extractions are going to be used for? Well, thank you for asking. Thank you for asking. Well, when they extract DNA, they are able to multiply it, and they are able to make they're able to use it, they're able to put it in PCR, and they're able to do molecular research. They're able to put it in PCR and then do genetic research. They can also do medical diagnosis. They can also do forensics. Yeah, and also it is frequently also used in biotechnology. Now I should be extracting. Yes, I can. Yes, I can. Yes, I can. No, yes, I can. But those strands are so thin, so it's like quite hard to take it out. Um, okay, let me see if I can take my. Oh my god, it worked, dude! It seriously worked, but I can't take it out because the strands are very less. It's even on here. It is on this fork. You see that? That is the DNA. So that is the thing that we are trying to achieve. Hey, you know what? At least I got it. Do you see that? This thing. That is also the DNA. So it's like clumps up like that. What is the DNA? So this is the DNA. You see how it's milky in color and it's not yellow? That means it's the DNA. So it's supposed to be like this, but it'll be in a thick strands. So if you use a lot of banana, it'll be more you'll get more. You'll be able to extract more DNA from the banana, but I only used one, so at least you can see it. That is the DNA. It did not come out in strands, it come out in clumps, but do not be worried, because you can try to do this experiment as many times as you want. I am just doing this to show that this actually works, and it does. So that is the DNA. I hope my professor accepts, because my camera is not really that good of a quality. It's an old tablet. But anyways, this is my successfully extracted DNA, my ancestors' DNA, and I hope. Oh yeah, before we end, I just want to say the reasons why it might be my DNA did not like come out in clumps and strumps. This is the DNA that I've extracted. I mean, yeah, there you go. This is the DNA that I've extracted, and the reason why I did not get much or get the stringy type of DNA that people usually get is because, might be one, because I did not use enough bananas. I only used one, as seen previously, or two, I used alcohol in the form of hand sanitizer. I did not use pure alcohol. Other than that, anything could have gone wrong. Maybe the hand sanitizer has a high amount of gel and glycerin instead of alcohol in it. It might be also because it's an old hand sanitizer that I've used 7 years. And also might be because of environmental factors. Maybe the lizard did something while I was turned away. Okay, that's not a valid truth, but it might be because like the alcohol, the hand sanitizer is not cold enough. But in this case, anything is possible. I hope this experiment successfully has demonstrated my ability to be a scientist. Thank you. But also, from this experiment, it has successfully showed that I have demonstrated DNA extraction using simple household materials. Like seriously, like that old sanitizer that your father kept in the cabinet. Yeah, you can use that to extract DNA. Oh, what? Oh, you're asking me about the DNA. Oh, there's even some on my hands. Oh, you're asking me about the DNA after you've done with it. Oh, you can just clean it normally. It's not radioactive, nor is it gonna ruin your household pipes. So just clean it normally as long as you take the goo out, because it's gonna be gooey. Dolly the sheep, I miss you, girl. Come back. I hope this is educational, and I hope this proves that I can actually extract DNA even at my home. Bye. Thank you for watching. Thank you.